At the meeting, we present the phase 1 first-in-human trial of CC-93269. This is a new T-cell engager, a 2+1 monoclonal antibody that's IgG-based. It has two domains that bind to BCMA in the multiple myeloma cells, and one domain that binds on CD3 on the T cells, therefore forcing T cells activation and engagement of the T cells into myeloma and causing myeloma cell death. This study was a dose-finding study. We start with a very minuscule dose in a single patient cohort, and we escalate all the way up to 10 milligrams. And we're now currently testing different ways to get to 10 milligrams with a step-up dose. The important thing is that the therapy was relatively safe considering this patient population. We had cytokine release syndrome, which is the hallmark of all the T-cell activating therapies, including CAR T cells. That was present in about three-quarters of the patients, but in the majority of cases was minor, grade 1 and grade 2, and responded well to therapy. We had, unfortunately, one episode of death from CRS on a patient who had a lot of disease and had a concurrent infection. The good news is as the dose increased, the responses became more common, quicker, and more profound. At the dose of 10 milligrams, 89% of the patients responded, and 44% of the patients had a complete response, and all the complete responses were also MRD negative responses. Overall, the study had response of 43% and 17%, but that includes the patients who receive lower dose of therapy. So we think this is a very promising agent, and we look forward to continue to accrue on this study that is ongoing and testing in slightly different schedules and doses, and we look forward to the future development of this drug.